So the Human Protein Atlas, it's been around for a while. It was started in 2003, headed by Matthias, as Angus said. And we are mapping the human proteome and trying to understand how our proteins are organized into these different functional entities. And we're a lot of people. We're about 100 people working in the project, and we've been about 100 people for this whole period of time. And the key to everything that we do is our in-house generated resource of reagents. So we have antigens, about 50,000 antigens, protein epitope signature tags that can be used to make antibodies and also used for different assays, such as targeted proteomics. And then we've generated antibodies, and about half of these, 25,000, have passed our quality control steps, and they can be used in a variety of assays. And now we're also producing full-length secreted proteins from mammalian cells in order to be able to do more functional assays. And everything that we do is very dependent on that our antibodies are good. So it's very important, as we heard yesterday, to validate your antibodies. And we spend a lot of time and a lot of effort on doing this. So I'm not going to talk about it today, but if you want to troubleshoot your antibody assays, we'll be happy to help out. We spend a lot of time on this. So another thing that we spend a lot of time on is actually the automation and the scaling up the assays, creating robust pipelines and standardized pipelines, and the collection of the metadata and everything that comes with it. So our key technologies are immunohistochemistry, immunofluorescence, transcriptomics, and targeted proteomics today. And the most important type of data that we generated is images. So we have about 20 million images in total, which half are publicly available. It's the largest collection that we know of annotated images, manually annotated images that shows either protein expression in tissues or protein expression in cells at a subcellular level. So all of these images, they're kind of packed into our database. So it's basically a catalog of images. It's open access. Version 18 is currently public. It's presented in a gene-centric way, and we have about 200,000 visitors per month. And we have these three different parts, the tissue atlas, the cell atlas, and the pathology atlas. So I thought I will briefly go through these milestone publications and the data that we have in these atlases, and then I'm going to move into how we can use this massive resource of images to actually try to understand biology. So starting with the tissue atlas. So the aim here is to map the protein distribution to specific cell types of all major organs and define the organ proteomes, and investigate how tissue-specific and kind of housekeeping proteins assemble to make our organs. And this is the first publication that was published a couple of years back, and here we've determined the expression profiles for about 17,000 proteins, and defined 13 organ proteomes, and the data is a lot of, about 3 million immunohistochemical images and a lot of RNA-seq data. So I'm not going to go into details. And we use tissue microarrays for these types of studies. It's 1 millimeter core sample. So when you think of it, it's very small regions that we're actually sampling. And the images looks like this. So you have the protein of interest that shows in brown. So it's a bright field image. It's an RGB image. The protein of interest shows in brown, and you can see a single cell resolution. So this would be CD44 that is expressed in the ciliated cells lining the fallopian tube. So you can see that this protein is not expressed in every cell, but in a certain pattern, every other or third cell. But that would be the conclusion from this image. And the conclusions from this study is that about half of our genes are expressed in all tissues of the human body, so that would be the housekeeping proportion. Many of the currently available drug targets, or the FDA-approved drug targets, are actually expressed everywhere and would be classified as housekeeping genes. And there's quite few proteins that are unique for one tissue, and less than 4,000 proteins are enriched in one or several of the tissues and organs. But these enriched proteins do make functional sense. So we find the transport proteins in the kidney and so on. So that's at least relieving. And then if we have a look at the organ with the highest number of uniquely expressed genes, the TESTIS, we can just, to illustrate how this data that is generated on the RNA-seq can be combined with the image data. So here we can have a look at these images of all the different TESTIS-specific proteins, and we can determine the cell type in which they are expressed. So the classification here would be that these proteins are expressed in spermatogonia, and these in the spermatocytes, these in the spermatid, and these in the sperm. So we can get a quite detailed cell type specific classification, but this is where the tissue atlas ends now, and I'll get back to that later. And then the most recent edition is the pathology atlas, the protein Atlas, and it was published in 2017. And here we want to investigate how protein expression is altered in cancer samples compared to normal, and identify genes with potential prognostic implication. And this work is done by Fredrik Pontian and Adil Mardinoglu at KTH. And the approach here was to use publicly available data from the Cancer Genome Atlas and integrate it with the HPA, RNA, and protein data, and try to analyze the major cancer types. That's 17 different cancer types, a lot of patients, and combining the coverage that they have in the TCGA, which is many, many patients, with the high-resolution protein images that we have in HPA, which is very few samples. We have about 20 samples of each cancer, and that covers all stages. So it's a very sparse sampling. So the data in the pathology atlas is about 7 million immunohistochemical images and 900,000 survival plots. So the conclusion of this study is that we could identify prognostic genes. So that would be genes that where a high or a low expression can stratify the patients into a favorable or unfavorable prognosis. So that's quite a lot. And no single gene did have a prognostic power in more than 7 cancers. So there's no universal prognostic gene, at least as we could identify. We could, again, demonstrate the heterogeneity of tumors, as many others have done, and that the heterogeneity within a tumor type can sometimes exceed heterogeneity between cancer types. And then we could also look at the proteins that we classified as being tissue-specific in the tissue atlas, and then we looked at those in the cancer pathology atlas, and we could see that they were significantly downregulated, as shown in this plot, and also demonstrated in the protein level here. So this supports the theory of dedifferentiation, that cancers are dedifferentiated as they become more and more malignant. So I'm just going to show you an example of the prognostic genes, because I will get back to this later too. So this is the kind of Kaplan-Meier plot that we present. In this case, this is timeless. It's a known protein. So it shows that a high expression is correlated to a shorter survival. So but it's very, of course, crude data, but this can be used as a starting point. So if we, for example, can see a similar correlation, we can see that patient with a low degree of the cancer has a low expression, and then with a worse prognosis, they have a higher expression of the protein, then that's at least a good starting point that this protein might actually be relevant for this cancer type. So I'll get back to that later too. But of course, this is half of all human genes, so it's a high number, and it should be considered a starting point for further studies. So with that, I want to get to the cell atlas, which is the part that I'm working mostly with. So I'm going to talk a bit more about this. So here we want to map the distribution of proteins and time and space at a subcellular level. And this is important because this is something that cannot be inferred reliably today from RNA sequences. Although I know that there's people here that are working on making that possible. So we want to define the organelle proteomes and investigate how protein spatial distribution contributes to cell functionality and cell type specificity. And we're doing this with fluorescent imaging, and I'm going to show you a lot of images of the different structures that we're currently mapping, and we're always using some markers. They will also be important to remember for later parts of my talk. So we use the DOPI for the nucleus, the DNA, microtubules to outline the cell, and a membranous marker too for the endoplasmic reticulum. So this is our 3 landmarks, and on top of that we add the HPA antibodies to study the human proteome, one at a time. So this is CD44 in the plasma membrane of the cell. If it looks like this, we can say this is a cytoplasmic protein. Actually, it's often ribosomal protein when we see these distinct holes here. Mitochondrial protein, the Golgi apparatus, plasma membrane, membrane protrusions. We can see lipid droplets. There's specific signatures of them, like they're round with a hole in it, so we can actually identify them quite reliably. We can see endosomes, nucleoli, fibrillar center, nuclear speckles. But there are different types of nuclear speckles. They're a bit harder to differentiate from each other, so this is all grouped. Splicing speckles and, for example, chromatin binding proteins will still be merged into this category at this point. We can see nuclear bodies, different types. Here you saw nuclear bodies where we can see a few nuclear bodies in every cell. We can also see more nuclear bodies that are more frequent. So here it's also hard for us to tell which nuclear body it is without some further analysis. We can see the filaments, intermediate filaments, actin filaments, focal adhesion sites, the centrosome. We can also see more rare events like the mitotic spindle. Proteins only localized to the mitotic spindle. What else do we have? Oh yeah, and this one I think is nice. We can see this is two very rare events, but we get a lot of biological information in this image. So this protein is found in the centromere in this dividing cell, and then it relocalizes to the cytokinetic bridge at the end of cell division here. So that gives us a very kind of, just one image provides a lot of information about the functions that this protein might be involved in. We can see dynamic structures such as the rods and rings that are formed when the glutamine homeostasis in the cell is disrupted somehow. And altogether, we've localized 12,000 proteins to 32 different cellular structures, and half of these proteins were not localized before. And we've been able to define 14 organelle proteomes. And 12,000 is still pretty far from the entire human proteome, but it's what we can find in cultivated cell lines, more or less. We need to find a different source of tissue or cells in order to map the rest. So data here, it's many fewer images, but about 400,000 high-resolution confocal images. A lot of RNA-seq data of a big cell line panel, and then also some GFP tag data and other types of tailored assays that I'm not going to go into today. So you heard Catherine talking yesterday, so we've, of course, love to compare our data with the other type of spatial proteomics methods. So for example, we've been comparing it with Catherine's data that she generated on the U2S cell line, and it was a very surprisingly high agreement, even though, for example, they're much better at resolving the vesicular structures and things like that. So there's a lot to win by augmenting these two data types. So about 80 to 90% agreement with LOPIT, and we've also done a larger study where we compared antibody-based localization with GFP-based localization, and again, there we had about 80% agreement. So it seems to be, there's disadvantages and advantages with all methods, but at least there's, it's nice that the core of the data is validated by several different approaches. And we've tried to define the organelle proteomes, and I just want to show this to point out that the number of proteins in each organelle is very correlated to the volume of that organelle. So of course you have most proteins in the nucleus and the cytosol, and there's most likely also a lot of, in the nucleus, for example, a lot of substructures that we haven't mapped at this point. But I will get back to this later again. So there's a lot of things that you can do with this data, but one thing that I think is pretty easy to do is to think of it as that there's different compartments of the cell, and of course proteins can't meet if they're not in the same compartment. So just add it to filters whenever you do some kind of systems biology study or filter interaction data sets and so on. So we did this with one data set just to show that we could remove about 20 to 30% of potential false interaction, and while doing this we could reveal completely different kind of hub proteins for the interactions that made more functional sense and had more central functions in the different organelles. So we think that this data can be used in this way, can be used and combined with transcriptomics and other proteomics data sets. And everything that I just showed you, that was a very, very short introduction to the data that we have in the protein atlas. So it's a lot of data, but we haven't really interpreted, or there's a lot of information about single protein one at a time, but we haven't really integrated it into some kind of whole proteome understanding of the human cell. So I'm going to tell you a little bit about our ongoing work, which bits and pieces that we're digging further into and what we're trying to find out, and some interesting observations that we've made. So I'm more focused on that kind of spatial single cell biology aspect of this, both in terms of the tissue images and in terms of the cell atlas images. So I decided to call this section spatial single cell biology. So one interesting observation is that a lot of the proteins are localized to several places in the cell. This protein is found in the nucleus and on the plasma membrane. This protein is found in the nucleoli and the mitochondria. This protein is found in the Golgi apparatus and in the nucleus. So in fact, half of all proteins localize to multiple compartments, and this is validated by Catherine's data too. And it's a bit surprising, but when we started reading up old studies, we could actually see that in the old yeast localization data set from Jonathan Weissman's lab, he actually reported the same numbers too. So it seems like about half of both the yeast and the human proteomes are located to multiple compartments. And we do see distinct patterns of this multilocalization that I think some are very interesting at least. So here's a plot that we try to show this. So here you have the compartments in the nucleus, and the cytosol, or the cytoplasm, and the secretory pathway. So if we start to look at the proteins that are found in structures within the cytoplasm, within the metacompartment of the cytoplasm, we can see that a lot of the components in the filaments, for example, are also found in the cytosol. This is not surprising. Proteins shuttle on and off, and they're associated to organelles and so on. So that's kind of not a surprise. Same thing in the nucleus. A lot of proteins that are in the speckles or in the nucleoli are also found in the nucleoplasm. That's not surprising either, but we don't see a lot of crosstalks between the different organelles in these two metacompartments. Then in the secretory pathway, we see more of a sequential kind of interaction pathway between the organelles, so that probably reflects the direction of traffic through the secretory pathway. But this is all makes sense, but it starts to get interesting when we look at interaction between these metacompartments. So it means things that are not actually in physical contact. So for example, the cytoplasm and the nucleus, of course, we see a lot of proteins that are in the nucleoplasm and in the cytosol, and a lot of nuclear receptors and shuttling proteins. Probably we see a lot of variation between cell lines also of the ratio of these nuclear and cytoplasmic levels, for example. And then we see a combination of different organelles there. It's not nothing super interesting there either. But then this is something that we find very, very interesting, that a lot of the components of the secretory pathway are actually also found in the nucleus. And we don't know what the functional connection between these organelles are, but this is, even if parts of it is unspecific antibodies, it's too much to be explained by that. So this is something that we're working further with to try to understand the functional connection between the Golgi apparatus and the nucleus. So we've started out by validating a lot of antibodies with knockouts, and it seems like there's a global pattern that we're trying to reveal, but I'll save that for the next time when we have some more data to back it up. So I think this is interesting because multilocalizing proteins may have a context-specific function and moonlight in different parts of the cell. So this would increase the functionality of the proteome quite a bit, and also the complexity of the cell from a systems perspective. And then we have other images that looks like this. So this is, for example, we see single cell variations. So this protein is only expressed in a subset of the cells. Sorry, I have to get some. And these cells are cultivated. They're in the log phase growth. They're not synchronized in any way, but they're clonal cell lines. We can see variations like this, more gradual variation, where we have high and low expressing cells. We can see translocations, where we have a protein that is found in the Golgi apparatus in some cells and in the endoplasmic reticulum in other cells. And in this image, you can also maybe get an hypothesis for the reason for this variation. So these cells, you can see that they're still attached. They just divided. So maybe a lot of these variations would be due to the cell cycle position. That was our initial hypothesis, at least. So we've been working a lot to get more data for the proteins that show single cell variation. We can see a lot of different patterns of single cell variability. We can see bimodal expression patterns and quite Gaussian distributions, and then some proteins that are expressed, not expressed at all, but then expressed at very, very high levels in a few cells. So there's a great variability in terms of the pattern of variability. And about 16% of all proteins show such single cell variations. So we can, just by looking at the images, group them into different types. So we have examples that show variation in the level of expression, on and off expression. We have examples that show a spatial redistribution, cytoplasm to nucleus, or mitochondria to nucleoli, or just, for example, the pattern within the nucleus. And then we have some proteins that we have a direct connection to the cell cycle because they appear in these cell cycle related structures, such as the mitotic chromosome or the cytokinetic bridge or the mitotic spindle, for example. So they're the easiest one to start with and confirm that they're actually correlated to the cell cycle. So we've done a larger study to test the hypotheses. Are these proteins expressed in correlation to cell cycle progression? And for this, we used a Fuji cell line that changes color, so it's red in G1, and then it turns yellow at the transition to S, and then it's green. And we use this cell line, and so this is looks like a FACS plot, but it's based on the microscopy images. So we can kind of see that this is the red population, so this would be G1, this is S, and this is G2. So we can isolate these cells and analyze them separately. So we've been staining this cell line with all the different proteins that show all the different antibodies that target these 16% of the proteome. And we can divide them into proteins that show correlation to the cell cycle, statistically significant, like this one. You can see that it's lowly expressed in the beginning and the expression gradually increases, or proteins where the variation do not correlate to the cell cycle. So it turns out that we were actually wrong. The majority are not correlated to the cell cycle. About 1/3 of all these proteins show expression correlated to the cell cycle. But it's an interesting group of proteins. They're mainly found in the nucleus, nucleoli, the mitotic structures, and the cytosol. If we do gene set enrichment analysis, we get functions related to cell division, DNA replication, chromosome organization, and so on. Less than 200 of these have a known connection to the cell cycle if we look at gene ontology annotations. But if we look in the pathology atlas and check how many of these do have a prognostic value in the pathology atlas, we see that as much as 82% do, and the majority of them are unfavorable. So it seems like it's an interesting group of proteins to look further at. We can also cluster the proteins based on the peak of expression. So we can see that the largest group of proteins are actually, so red is the highest expression, are actually found in the late phases of the cell cycle. A smaller group is peaking in the kind of S transition, and then a larger group of proteins, again, are found in peaking in the G1. And then if we look at where these proteins are found in the cells, we can also see some patterns. So for example, we see that nuclear proteins, they're in all of these groups, whereas most of the cytosolic proteins, a lot of enzymes, they're peaking in G1. In the very first phase here, early G1, we see a peak of proteins in the nucleoli fibrillar center. We haven't really understood this connection yet. Maybe Angus can help us on that. And then in the proteins that are peaking in the S phase, we have an enrichment for proteins in nuclear speckles. And then towards the G2M phase, which is still not resolved very well, we have early on proteins in the nuclear membrane, and then these mitotic structures that would probably be expressed in the mitotic phase. So what is interesting here is that the majority of the proteins are peaking in G2. We've been trying to look at transcriptomic studies to compare it, and most of them report a peak kind of transcriptional burst, or more early on in the cell cycle in G1, but the overlap between our 551 proteins and the studies of gene expression correlated to cell cycle is very poor, so it's actually hard to make any conclusions. It's, so either it's, we haven't really explained this, either a lot of these proteins are regulated at the post-translational level. We also haven't investigated if these proteins are actually drivers of the cell cycle or if they're just passengers of the cell cycle. So that might be some of the explained part of the poor overlap with transcriptomic studies. So we're currently generating a single cell sequencing data set so we can actually check this with our own in-house generated data. Oh yes, and we do also get different functional enrichment in these different parts. If we take the proteins that are peaking in S, and it all makes functional sense. So there is some function embedded in all of this, even though there's a lot of uncharacterized proteins in here. And I just wanted to show two examples, because we can actually find a lot of interesting potential clinical markers for proliferation and cell cycle in here. And I'm just going to show two different examples. One is a protein that is correlated to the cell cycle. We've seen it in many cell lines. We have two different antibodies that give the same pattern. And a high expression is correlated to an unfavorable prognosis in several types of cancers. And we can also see that this is, for example, normal liver and liver cancer. And we see actually very selective expression that is only found in some cells in the liver cancer. So there's definitely proteins that we will follow up and study more in depth and see if we can use them as clinical markers for proliferation. There's also another example that is actually correlated to favorable prognosis. So there are some interesting examples like that. This is a zinc finger, also again, two antibodies that show exactly the same pattern, and it's correlated to favorable prognosis in 4 different types of cancers. No. You just have to watch this one a bit longer. I don't know. Oh, there we go. So in order to, this is still not a very highly resolved data set. You saw the big G2M bulk of proteins there. So in order to resolve the cell cycle better and be able to map the RNA expression versus the protein expression, we're developing a computational model to infer the cell cycle position from the reference markers in the images. And it's nearly done, but we have some batch-to-batch effects that we haven't figured out the best way to correct for yet. So this is an example of an image from the atlas. So we can just apply this machine learning method to these images, and then we can measure the intensity of the green staining and map it onto this linear trajectory of the cell cycle. So we can see that this protein KIF23 is actually peaking in early G2, and then it drops again. So by doing models like this, we hope that we can be able to both map the RNA expression and the protein expression to be able to correlate their expression and actually get a very highly resolved map of the proteome and the human cell cycle. And to get back to the majority of the proteins, the ones that are not correlated to the cell cycle, this is actually even more interesting in my opinion. So why do we have almost 800 proteins that show a clear on and off expression in cells, and it's reproducible in many different cell lines? I think that's a super interesting question. So there's some patterns that we can see for this set of proteins. So they are found in different parts of the cell. So it's also nucleus cytosol, but then we see a lot of proteins in the mitochondria and the secretory pathway and in the intermediate and the actin filaments. What is interesting here is that a lot of them, 73% of these genes, also have a prognostic value in the pathology atlas. But here it's equal parts favorable and unfavorable. So it also seems to be an important set of proteins. If we do a gene set enrichment analysis of these 800 proteins, we get no significant enrichment. So it points us to that this is probably related to a lot of different things, since we're not picking it up in the enrichment analysis test. So we're currently working on this. If you have any ideas, I'll be happy to hear them and discuss them. So these are just some examples of mitochondrial proteins, for example, SLCs, solute transporters, that show a clear on and off expression, not correlated to the cell cycle. Same thing with a lot of intermediate filament proteins, on and off expression. So what's the reason for this? There's many different hypotheses. It could be the cell confluency or the neighbors, the topology of the organization of the cells. It could be the metabolic state, or it could even be some kind of proteome bed hedging, or stochasticity. We don't know. So we're trying to explore this a bit further now. So with that, I would like to move over to image analysis, because there's a lot more we could understand from these images if we analyze them a bit more, rather than just saying this is a mitochondrial protein and it's found in half of the cells. That's a very crude analysis of the data. So we have 20 million images, and with all the developments in machine learning, this is kind of a rich resource for actually exploring biology. So that's what we're trying to do. So I'm going to give one example of how we're trying to go deeper in the tissue images. So in the examples that I showed you previously, we classified protein expression according to cell type. But what about the variability within a cell type? That's not classified right now, and I wouldn't want to look through 20 million images and do the classification manually. So this is an example of pancreas that we just started with for reasons, practical reasons. It's easy to work with computationally. Here you see the islets of Langerhans, and this is the exocrine cells. So this is an example of a protein that is shown in brown that is more or less expressed in all exocrine cells, okay? But this protein is also annotated as it's expressed in exocrine cells, but as you can see, it's not at all expressed in all cells. So it's a very different pattern. And it's actually kind of a regular pattern. So this protein is expressed in cells that are dispersed all over this area and in a kind of regular pattern. Whereas we have other examples where we see that this protein is not either expressed in all exocrine cells, but when it is expressed, it's expressed in all the cells within an acinar cluster. So we're trying to, well, I have one more example, I think. Yeah, and then we can also see examples like this, where this protein is expressed in maybe 25 cells in this whole field of view, not picked up at the RNA-seq bulk level, for example, because it's a rare cell. So we're trying to capture all of these variations, and the way that we want to do it is, of course, to try to avoid segmentation, because that's really hard, but see if we can generate computable metrics representing the protein expression topology in the tissue. So we're trying to find good metrics that can describe the dispersion of the expressing cells, for example, and see if we can predict protein co-expression by this. And then we could do the follow-up experiments and co-localize the proteins experimentally. And I should say that there's a lot of proteins. Just in pancreas, we have about a list of 500 proteins that show such single-cell variations within the exocrine cells. And then when it comes to the subcellular images, we have localized 12,000 proteins, but it's all separate images. So if we're going to claim that we're studying the human proteome, maybe we should actually try to integrate it somehow. So here we're trying to test models like GAN models or autoencoders to see if we can actually create an image of a canonical cell based on the input of the many images that we have. So that's the long-term goal, and we're right now trying to see which organelles can be predicted. Can the distribution of one organelle be predicted from the distribution of another organelle, and so on. But what we have done is that we've worked a lot with developing tools for recognition of the patterns in the images. So what I just presented to you is what we're currently trying to do, starting to do, and this is something that we have done. So there's a lot of patterns in the images that I showed you, many more than we're actually classifying at the moment. And you might think that this is a trivial task, but it's actually not a very trivial task, and there's many reasons for it. There's a great class imbalance that's important if you do machine learning. So we have very common patterns, the cytosol and the nucleus, a lot of proteins are found there, and we have a lot of rare patterns. Like we can have 5 images of a certain pattern, for example. Second challenge is the morphological difference. We have many different cell lines, and this is an example of an active filament protein in 3 different cell lines. They're visually very different. Also, the single cell variation, 16% of the proteome, and then the multilocalizing proteins, half of the proteome. So it's actually a multi-label question. So this is not a trivial task. And there's some good classifiers published today, but all of them are ignoring, all of them are focused on one cell line and single label location. So this is quite more complex than that, unfortunately. So we've developed a machine learning method that we call LOCAT, that can DNN train to classify the subcellular patterns. And I'm not going to go into the details. It performs well on some organelles. It performs less well on others. It can do the multi-labeling thing. And we can see, not surprisingly, that if we have a lot of training data, it performs better. That's the kind of general observation. So as a measure of accuracy, we're using the F1 score, harmonic mean of precision and recall. So it's about 0.65, the mean per class F1, so it's not perfect at all, but it's pretty good. It's something that is good enough to work with at least. So we're starting to implement this classifier into our workflow, and we're actually removing the manual layer of annotation. But when we train a machine learning method to recognize the patterns that we've annotated, we're actually not discovering anything new, right, if we want to find new patterns. So in parallel to this, we also did another approach, which was based on citizen science. And you probably know of a lot of nice gamified citizen science projects like Foldit, the first one I would say, and then 2 of my favorites, Eterna, where you fold RNA and predict, now I think they have a CRISPR effort ongoing, and then also iWire, where you map neurons in the brain. They're all wonderful games that produce high quality data. But all of these efforts have struggled with the recruitment of players to these games. They have to put a lot of effort on the actual outreach and trying to get people to participate. And we did a different version. I've told many of you about this before. So we integrated, in order to solve this, we thought that isn't it better to integrate the scientific task into an existing game that already has a large base of players and just tap into that? That would be a great resource for science. So we created a mini game for pattern recognition in microscope images together with Attila Sandner. It's this guy here. He was the first one who thought of the idea, and he's founded a company to do this kind of projects with the researchers and gaming companies, that MMOS. And then it's CCP Games on Iceland. They're a major producer of computer games. So together we developed something called Project Discovery, and it's integrated into the game EVE Online, which is a large sci-fi MMO online game with about 500,000 players and about 40,000 online at any time. And we had to sign off to that we couldn't interfere with how the game developers actually integrated this into the game. So I'm going to show you a movie now that was broadcasted in-game, in the in-game news channel when the game was launched. So this was March 2016.
The sudden drifter pullback to the 5 known hive systems are believed to be connected to the capsuleer reports that gate restrictions in those systems have been removed, clearing the path for capsuleers committed to pushing back the drifter menace. The conflict has certainly taken its toll on the Amarr military, and talks of the withdrawal being a reaction to Sisters of Eve, and capsuleer actions only adds insult to injury for the Amarr. Meanwhile, Sisters of Eve has launched a crowdsourced analysis of collected drifter data entitled Project Discovery and rallied under the banner of Citizen Science Begins With You. They hope to unlock the new drifter tech so that they may be made public and shared with all. This is Lena Amber reporting for The Scope.
So the game designer, the game developers tried to connect this to their story by integrating it into a kind of narrative with these characters called the drifters that were invading this universe, and in order to kind of counteract their invasion and understand them and maybe clone their superpowers, the more friendly in-game corporation called Sisters of Eve launched this crowdsourcing initiative in-game to map the drifter DNA, and this crowdsourcing effort was headed by Professor Lundberg in-game, of course. Immediate threat, and you rely on the scientists, right? So that's the kind of how they try to do it in the narrative, but the actual game is pretty simple. It's a small window that you can open at any time in the game. There's some information, there's biological information, and there's only kind of non-biological information. But it turns out that a lot of the players actually were interested in the biological information. We have never had as many hits on the dictionary pages of the Protein Atlas as when this game was launched. It increased with 20,000 a day. So that's nice. So we're trying to educate people about proteins. There's also a lot of other motivational things such as ranking. So you can start out as novice of analyst. You can end up as professor of analyst if you're very persistent and play for a long time. And you can also win project discovery specific swag such as combat suits, and you can sell them and you can make money in games. So the motivation for playing is a bit different than in the previous citizen science games because you actually earn money in game. But the results were still overwhelmingly good. This is tenfold higher than we had set up as our dream goal. So the project was open for 1 year, and we had 322,000 players playing the game. So it's clearly very efficient as trying to recruit players to the game. And they provided 32 million image classifications, so that's a lot of data. And they spent 70 working years if we do add up the time they spent on the minigame in the game. So it's I don't know, computer games is a great way to tap into people's brains for sure, and have them dedicate their time to something productive maybe. Let's see, because this is still the kind of nature of this game. So was this good? Can we produce data with high scientific quality compared to these other projects that actually have proven that they've done that? So there's a lot of different aspects here that I'm not going to go through. We have tutorial, we have training phases, we have control images, and different things that drive the participation. But we've analyzed the whole data set 5 times, and I'm just going to jump right to the conclusions. So to remind you, when we started this, we only classified 23 patterns. So we asked the players, in comparison to the DNN, to classify 23 existing patterns. So this is something we can use to measure their performance, but also 8 new patterns. And if we look at their performance, here we can see these are some random players. F1 score again, and the log 10 number of images that were analyzed, and every dot here is a player. So you can see that the average player is like 0.25, that's pretty bad. But the line here represents the power of the crowd, so that's the aggregated result. So you can really see that together it's actually pretty good. 0.55 almost. And we can also see, we can identify the players that didn't play seriously. They're the blue players here. So that's the players that only clicked cytoplasm nucleus, because of course they figure out that that's the most kind of greatest chance of winning the lottery. So we can see that those players, they don't play for very long and they actually get worse. Whereas the rest of the players, some of them played for a really long time and classified a lot of images. And they don't improve a lot, but they do improve a little bit when they've done a couple of hundred images, at least. And we can also measure the performance on a day-by-day basis. So we changed the game at some point, for example, here. And here we introduced a harder training set of images. So with artifacts and background staining and a lot of things that we hadn't included in the beginning. And then we can see that during the coming month, the players actually improved a lot in their performance. So there's things that we can do to kind of drive the performance of the players in this way. So if we do the same kind of map again and see how are the gamers doing per organelle, what are they good at, what are they bad at, they're kind of equally good as the machine learner, but we don't see the same correlation to the amount of training data, of course. So they can actually do very well on small sets of data where the DNN did not, for example. But overall, they're comparable. But what is interesting is that if we compare them organelle by organelle and ask that out of all actin filament images, how many did the DNN get right and how many did the players get right, we can see that it's not a perfect overlap. We can see the gamers, the green, they're much better at some things, like the microtubule organizing center, whereas the DNN is better for endoplasmic reticulum. But there's still not a perfect overlap. So it means that the gamers are capturing some type of information that our deep learning network is not catching up. So maybe we can actually improve that. And what are the gamers actually seeing that the network is not seeing? So this is something that we're trying to use the data set now together with other researchers at Stanford to look at the learning dynamics and understand what the gamers are seeing that the neural nets are not seeing. But in order to compare them, really compare them side by side, because that's the question that I always get, so who's better? Or is there any use of having gamers when you have AI? So in order to do that comparison, you also have to compare it to the experts. So we're quite confident in that we can do this good. So I blind tested 3 people in my lab. And luckily, they were at least beating the gamers. There was this one gamer who's actually up here. I should recruit him. But yeah. So this is recall and precision and the average of the 3 experts in my team. So this is the baseline of it. And this is, I should say, the mean per class precision and recall. So we're punishing, if you're good at the rare classes, it will help you in this comparison. So we can see that when we do the comparison this way, project discovery is slightly better than the DNN. If we look at the overall F1, it's the other way around. So then if we just take the annotations of project discovery and augment it into the deep neural network, it gets better. So that's nice. But on the other hand, that means that we would have to pass every image through the game in order to get that accuracy, and that's not really sustainable because the game is already closed. So then we trained another network to mimic the gamers. So it never even saw the ground truth of the images. So it just mimics the gamers. And then we use that to augment the original LOCAT classifier. And it's actually improving quite a bit for some of the rare classes. So now at least we have a classifier that performs pretty well. And it can also do this on new unseen images. So the conclusion is that we've shown 2 alternative approaches for large-scale image classification and a path forward to how you can combine MMO, citizen science, and machine learning. And ideally, you would probably build your citizen science projects so that it's integrated and developed in real time. And we are, but still, we would still like a classifier that could replace the experts or even beat the experts. So in order to do this, we're making the whole data set publicly available, and we're launching a public image challenge together with the CCI initiative. So that will be launched sometime this summer. So if you're interested, please give it a try and help us develop a better classifier. Before I leave that part, I just wanted to say that the gamers they did very well in finding rare patterns. So they helped us to identify another 30 rods and ring proteins, and also to find a lot of proteins that are specifically localized to the outer rim of the nucleoli. So they did add new biology to the protein atlas. And it did also give us a lot of outreach and attention to a crowd that we normally don't reach out to. As well, that it was a student of mine gave lectures in the in-game university. So it's a completely different channel of outreach, and that was very fun to explore and teach people that proteins are not only something that you eat, they do a lot of things, so that was fun. And also the feedback from the players. It was an amazing project to be part of. Yeah. So with that, I would like to move to some conclusions and future aims and give you the directions of the Protein Atlas moving forward. So first, we're still aiming for complete coverage, which will maybe turn out difficult. So one important thing in order to get to complete coverage is increased sample diversity. So we're gonna include more specialized tissues such as lens or fetal tissue, hair, lactating breasts, glands that we're missing, work with different types of cells like stem cells and primary cells, and also include plasma samples to find the secreted proteins as well. We're adding new chapters, and with chapters I mean these kind of silos of the Protein Atlas. So we're adding a blood atlas later this year, and then the mouse brain atlas next year, metabolic atlas in 2020, and then also a developmental atlas is on the roadmap. And we're also going to try to merge these atlases a bit more and break the silos and actually try to model cell types, for example, and integrate different types of data and hopefully go towards some kind of functional understanding of the different cell types of the human body. So my main conclusions of this talk would be that we're trying to map the building blocks of humans systematically in cells, tissues, and cancers. It's an open access resource for scientists. And well, we know that a lot of you are using it, and we're very grateful for it. And all input, if you have feedback for what you would like to see, please don't hesitate to tell us. We demonstrated a complex cellular architecture with many proteins that are expressed in all cell types. We see multilocalizing proteins variation between cell types and also single cell variations. Science and gaming is a very powerful combination, and we're looking into ways of exploring that further. And the aim for my work would be to provide a proteome-wide integrated model of a canonical human cell over the course of one cell cycle. So hopefully one day I'll get there. And with that, I want to thank This is the Cell Atlas team and collaborators such as Catherine and the developers of EVE Online, and also the funders. Thank you.
You probably didn't capture this, but have you got any idea of the average age of your players?
Yes, we do. I think it was 39. 95% male. But what is very interesting is that 95% of the players are male, but 50% of the characters are female.
Oh.
That's an interesting...
Is there anything you can tell us about the range of characters that were personified?
No. Actually, we don't have access to too much of that information due to restrictions in the EULA when they enter the game.
There's so much information in there that too many questions to ask. But from your single cell differences, I'm wondering, as you pointed out, going forward, there are so many functional and biological questions that you can address. So I'm wondering if there's information embedded in there about cellular communication. Let's say one cell having an impact on neighboring population, and you see a response from the same protein or something in the other ones. And how this can connect going forward, let's say, with cellular pathogen interaction or microbiome and things like this, right? So that's one question, and then I'll have one other quick one.
Okay, can I answer first? Or
yes, sure.
Yeah, so that's a very good question, and we're just at the early start of it. So right now we're definitely thinking that the topology, so that would be the kind of connections to the neighboring cells, but also connections to other cells, indirect connections. So right now we're looking at models from ecology, actually, where they look at animals in big kind of reserves and how they move in relation to each other. So we're trying to apply some of those models into, at least to get some kind of quantitative metrics of the topology. That's, I think, the best starting point we can do. And if we can narrow it down to some set of 30 proteins that we can multiplex, then we can do quantitative assays from there. But I think it would be very nice to have that kind of topological scaffold that you can use, topological metrics that you can use as a scaffold for integration of other types of data. That's what we're trying to get to.
And the other question I had, so in moving forward, if you're now going to introduce, let's say, all these different perturbations or trying to find out how things change, right? and the dynamics and temporality or things, it gets quite complex. So how rapid is this image classification, and can you actually take these signatures and do something like face recognition, kind of similar to what's done for that, and use it so that you can quickly process images, right?
Yeah. So that's a good point. The classification is very rapid, so that's not a problem. And there are good, for example, Google has a bioarchive publication where they took their facial recognition algorithm and apply it on recognizing cellular pattern, and it does very well. And we're actually going to try it. We just gave them a data set, and they're going to see if they can recognize the tissue type, for example. That's a simple thing. So we're actually trying that now, because it should be possible. What was there another? No, yeah.
Matthias?
I had a question to the underlying resource of your antibodies. So is that just frozen and done, or are you developing that further, or making monoclonals out of them?
No, at the peak we did 600 antibodies a month, and now we're doing 200 antibodies a year. So it's not a lot, and we reason that if we made 4 attempts for a gene, and we've made at least 4 different antigens, and we immunized all of them at least once, and we didn't make a good antibody, then either the type of antigen is not the right way, or the host, should be some other type of binder. Maybe you need the PTMs to get a good binder. So we're reasoning that then we're not using the right approach. So right now we're doing a call for, so when we started the project, we had a call for and took all the 13,000 commercial antibodies and tested them. And then there's been a lot of commercial antibodies developed during this period. So now we're making a call for new commercial antibodies for all the genes where we don't have a high validation already. So that's our approach at the moment.
Just before Tuna gets the microphone, so if I can have a question. Yes, I can. Right. So this is a fantastic resource and piece of work, and obviously throws up a lot of questions you can delve into in different ways. When you're showing the general conclusion that many proteins exist in more than one location, how can you be sure it's really the same protein? I think Neil isn't here, but if he was here, he'd be saying proteoform, and how do you know you've got the resolution already to actually say that's the same protein as opposed to an immunologically reactive protein? It might be an isoform from the same gene, it might be a process form, or whatever.
Yes, so that's a very good point. So we don't have the resolution to make that distinction, and most of our antibodies are targeting at least the majority of the splice isoforms, if we just look where the antigen is. So we can't make that statement. When I talk about proteins, I mean one representative protein per gene. So we're currently trying to see if we can look at the proteins that show differential distribution between 2 cell types, and then look at the RNA-seq data and see how much could splicing account for this difference, for example. So that's the only thing that we're looking at the moment to try to explain that.
So from Sarah's comment earlier, it's more of a protein smoothie you're looking at.
Yeah, exactly.
And so just following on from it, have you thought of doing the experiments, I think to what I was showing, where you could maybe take some of the cells, for example, that show differential expression of proteins, you could then sort those proteins when you've stained them and actually compare the population at the proteome level between those that are expressing a particular protein and those that aren't. It would be quite interesting to see if there's some other feature there that distinguishes these. Because what you're doing is providing markers for subpopulations, and then there's a method now to pull them out and actually see what the differences are.
Yeah, so we would love to do that.
My question just follows what Angus asked about this multilocalizing proteins. So half of the human proteins localized to multi-compartments or subcellular localization. And the idea is that every protein in a cell communicates with something else to make a function. And of course, if they're localized to multiple compartments, then they should be making complexes that are different for different compartments. And their question is, have you tried to overlap those multilocalizing proteins to protein complexes they associate with, for example, the SAC complexes, or from the lopit, or unclad Gown said, from these, all these complex basically that are associated with, if you overlap, can you say that these proteins which are multilocalizing, they have a different complexes because the complex members unlikely localize in the same way to different compartments?
Yes, so no, we have not done that investigation. We don't have I don't know the resolution, or maybe we could do it now with combining with some other data sets. It would be a very interesting comparison, because I agree with you, because that would be the hypothesis behind it all, that the protein meet different proteins in the different compartments, so we should find different complexes.
Particularly if you find the complexes that would support your notion that the protein really has different functions, different compartments. If you don't, then at least then the caveat of whether antibody could be, having issues, at least you can help to sort those issues out.
Yeah, so we've done this, for example, for 50 proteins that are found in the nucleus and the Golgi apparatus. We've done it for a few proteins that are in the vesicles and the nucleus. So there we've demonstrated it, but it's still kind of random examples to make a global statement. So we've proven it only for a certain number of handful of proteins.
If you take some of those proteins that are variable and you express them under heterologous promoter, are they still variable in their protein amount?
I don't know, we haven't done that experiment.
So I was curious to understand a bit more about whether there is scope to analyze or to identify sort of cell states in terms of their morphology and their structural position within a tissue. Because you showed the fallopian tube, for instance, and that sort of punctate pattern every second or third cell on the surface, essentially, of the structure. I mean, that's obvious. That's kind of another level of machine learning or image processing problem, right? And are there any? I mean, I don't know the field, but are there any attempts to kind of go into that?
To analyze the pattern, that's kind of what we're trying to do, the topological patterns. The big challenge here would be that these are terrible images to work with, because it's RGB images, and the brown color changes spectral properties depending on if it's lightly brown or dark brown. So it's a very hard, it's a hard type of images to work with. So that would be the major challenges. And besides that, I think in the field.
So the main game thing that you showed was more about the subcellular.
That's more classified,
yeah. But what I meant was, so taking a fallopian tube or decidua or whatever, skin, section and so on and so forth, and then trying to identify the differences in the morphologies or and relate the protein expression to that, and then superimpose.
Yes, so the reason that we're going for the topology instead of that is to be able to do it without the segmentation, because solving the segmentation, that's a hard problem. But a lot of people are working on it.
So the cell individual cells.
Yeah, so if you really want to analyze that type of expression in the cells and quantify what you're talking about, then you would have to segment the cells.
Yeah,
and
correct.
that's a hard thing to do. Yeah.
Okay.